Shah, and today we will discuss microbiology. The book name is Tortora, and chapter 1, part 1, the microbial world and you. So microbes in our lives. Microorganisms are organisms that are too small to be seen with the unaided eye. Germs refer to a rapidly growing cell. Microbes are of two types, good and bad. Good, which decomposes organic waste, are producers in the ecosystems by photosynthesis, produce industrial chemicals such as ethyl alcohol and acetone, produce fermented food such as vinegar, cheese and bread, while bads are those which cause diseases and thus called pathogens or pathogenic. The naming and classifying microorganisms. Linnaeus established the system of scientific nomenclature. Each organism has two names, the genus and specific epithet. Scientific names are italicized or underlined. The genus is capitalized and the specific epithet is lowercase. Scientific names are Latinized and used worldwide. May be descriptive or honor a scientist. Descriptive means to describe its characteristic, the microbe characteristics, and honor means to honor the scientist who discovered or to give some honor. Staphylococcus aureus describes the clustered arrangement of the cells, staphylo, and the golden color of the colonies. Scientific name Escherichia coli honors the discoverer Theodor Escherichia and describes the bacterium habitat, the large intestine or colon. Scientific names after the first use. Scientific names may be abbreviated with the first letter of genus and the specific epithet. Staphylococcus aureus and Escherichia coli are found in the human body. S. aureus is on skin and E. coli in the large intestine. These are some pictures of microorganisms with different shape. First, we can see on left that is bacteria, and it is bacillus because it is in rod shaped. And then sporangia, food particles, pseudopods, and different viruses, bacteria and fungi and algae are shown in these pics. Bacteria. Prokaryotes, peptidoglycan cell walls, binary fission, for energy use organic chemicals, inorganic chemicals, or photosynthesis. So these are the characteristics of prokaryotes. Especially we are talking about bacteria. Archaea are prokaryotic, lack peptidoglycan, live in extreme environments, include methanogens which produce methane gas, extreme halophiles which lives in extreme salt places where a high level of salts are present. Extreme thermophiles means that they are present, they live or they tolerate extreme level of heat. Fungi are eukaryotes. Their cell wall is made of chitin. They use organic chemicals for energy. Molds and mushrooms are multicellular, consisting of masses of mycelia which are composed of filaments called hypha, as we can see in the picture. On that mycelia or hyphae we can see sporangia, which is the reproductive part. Yeast are unicellular. Yeast are also fungi but are unicellular. Protozoa are eukaryotes, absorb or ingest organic chemicals. May be motile via pseudopods, cilia or flagella, as can be seen in the pic. Algae are also eukaryotes, cellulose cell wall, use photosynthesis for energy. We can see the greenish color. Produce molecular oxygen and organic compounds. Viruses are acellular, consist of DNA or RNA core. So its core may have RNA or DNA, like HIV virus contain only RNA, or others may contain DNA. So it is viruses are something which may contain RNA or DNA. Core is surrounded by a protein coat. Coat may be enclosed in a lipid envelope. Viruses are replicated only when they are in a living host cell. So they are not viable outside the host body. So that's why we called it that viruses are on the boundary of living and non-living organism, because they do not behave viable outside the host body. Multicellular animal parasites. These are the parasites like helminths. Eukaryote, they are eukaryotic. So I must tell you here that eukaryotes are those which can have a true nucleus, means have cell membrane, while prokaryotes do not have their not cell membrane, the nuclear membrane. Eukaryotes are those which have nuclear membrane around its nuclear material, while prokaryote does not have any nuclear membrane around its nuclear material, and the nuclear material is just aggregated in center or some a bit more sidewards. So it doesn't have true nucleus, the prokaryote, but eukaryote have true nucleus. So they are multicellular animals. Parasitic flatworms and roundworms are called helminths. Parasitic flatworms and roundworms are called helminths. Microscopic stages in life cycles. Classification of microorganisms: 3 domains: bacteria, archaea, and eukarya. So in eukarya comes protists, fungi, plants, animals. The first observation happened in 1665 by Robert, who reported that living things were composed of little boxes or cells. In 1858, Rudolf Virchow said cells arise from pre-existing cells. Cell theory comes: all living things are composed of cells and come from pre-existing cells. The first observation was made by Antony von Leeuwenhoek in 1673 to 1723. In between that, he observed by his handmade microscope that living organisms that he observed in teeth, scrapings, rainwater, and peppercorn infusions. The debate over spontaneous generation began. The hypothesis that living organisms arise from non-living matter is called spontaneous generation. According to spontaneous generation, a vital force forms life. The alternative hypothesis that the living organism arise from pre-existing life is called biogenesis. So for proving their hypothesis, there were some experiments done by Francisco Redi, who filled 6 jars with decaying meat. The conditions are written in the table: that 3 jars covered with fine net and 3 jars open. So the jar which was covered with fine net, there no maggots were grown. In other case, maggots were appeared. From where did the maggots come? What was the purpose of the sealed jar? Spontaneous generation or biogenesis? In 1745, John Needham put boiled nutrient broth into covered flask. So condition was that nutrient broth heated, then placed in sealed flask, microbial growth occurred. From where did the microbes come, and spontaneous generation or biogenesis? Let's discuss another experiment which occurred in which was carried out in 1765 by Desiro Spallanzani, who boiled nutrient solution in flasks. Condition was that nutrient broth placed in flask, heated, then sealed, so found no microbial growth, spontaneous generation or biogenesis. Louis Pasteur demonstrated that in 1861 microorganisms are present in the air, so nutrient broth placed in flask, heated, not sealed, microbial growth occurred. Nutrient broth placed in flask, heated, then sealed, no microbial growth, so hence proved. Pasteur S-shaped flask kept microbes out but let air in, so the experiment proved. The golden age of microbiology. So we will start from here in our next video. Please subscribe and like my channel and share it with your friends. Thank you, and your suggestions will be highly appreciated. Thank you.